Hi again, everyone. So we're gonna talk in this second part of the video that I made for the gas exchange about the transport of gases, and when it comes to the blood and the gases we are transporting, we're really talking about oxygen and carbon dioxide. Well, also we need to realize that nothing is random. We have to have a superior control over the amount of oxygen that you load on your blood and unload to the tissues. And the same thing, you will have to have control over the amount of carbon dioxide that you load and unload. And there has to be a relation of some sort to tell you that if you have too much carbon dioxide, it's really the good time now to give up your oxygen because the tissue needs it, right? You will also realize if there is increased acidity of the tissue, that means the tissue is metabolically active, and that's a really good time to give up your oxygen because otherwise you're going into glycolysis, and accumulation of hydrogen ions are not necessarily a good idea for the tissue. So conversely, you will also realize that when you oxygenate your blood, it has to send a signal to the carbon dioxide to come off. And that happens at the lung level because now your blood is oxygenated, and you don't want the blood, now that it's oxygenated, to go around and keep circulating with its carbon dioxide. You want to encourage the blood, the hemoglobin in this case, and other components of the blood to give up its carbon dioxide. So there has to be this kind of intrinsic relationship between oxygen and carbon dioxide, between oxygen and hydrogen ions, and vice versa. You have to have that, and we will see how that is controlled by two factors that we will talk about. We call that the Haldane effect and Bohr effect. And those are very, very important effects that we will have to deal with later on. Okay, so initially, first thing first, we are transporting gas, and the blood is the important vehicle that you are transporting oxygen, carbon dioxide to and from the lungs. Got that. We will realize together that oxygen is very poorly dissolved in water, very poorly dissolved in your plasma. So it really has to be carried as oxyhemoglobin. It binds to the hemoglobin, but once it binds to the hemoglobin, as you can see here in this equation, once it binds to the hemoglobin, that oxygen is not available for the tissues. You have to realize that the tissues are able to consume and play with the free oxygen, not the oxygen that is bound to the hemoglobin. That is not available, but it will become available if the oxygen concentration drops. Then the oxyhemoglobin is going to give up some of its oxygen. So you have to realize that the oxyhemoglobin is only a vehicle, right, to carry the oxygen, right? On the other hand, also, because this oxygen, the dissolved oxygen, is the only one that contributes to the concentration. Remember, this is kind of sequestered oxygen. So it doesn't really contribute to the concentration of oxygen in the blood or in the plasma. And we agreed earlier that the concentration of blood in a solution, right, we will call that partial pressure. So it's the dissolved amount of oxygen in the plasma that's the only one that's contributing to the partial pressure. And obviously, if this goes higher, then the equation is going to this direction, to the right. We studied that in chemistry at some point. If the oxygen is dropping, because this is reversible reaction, then it's the opposite. So if I increase the oxygen, then I make more oxyhemoglobin. If I reduce the free oxygen that's in the plasma, so if I reduce the partial pressure for oxygen, then the oxyhemoglobin is going to dissociate and gives me free oxygen. That's a very important part. When we get to the oxygen dissociation curve, it's all based on this phenomena. The fact that if there is higher amount of oxygen, free oxygen, then you are loading the oxygen on the vehicle, on the oxyhemoglobin. Once the free oxygen drops, then the oxyhemoglobin will cough up some of its oxygen to be utilized by the tissue. Keep that in mind. So, this is what I pretty much explained to you. It's a very small percentage of oxygen that's dissolved in the plasma, very small, 1.5% of the oxygen is dissolved. The rest is binding to hemoglobin, giving you what we call oxyhemoglobin. And the oxyhemoglobin is not necessarily participating at all in your partial pressure for oxygen. Keep that in mind. So, how do we carry the gases? We're only talking about oxygen and carbon dioxide here. Remember, nitrogen doesn't really dissolve well, and we don't have a vehicle for it, and the only time it dissolves in our blood is when we are under a high pressure, like diver or of some sort. So, but we're dealing here with oxygen and carbon dioxide. So let's talk about oxygen first. So oxygen is, very little of it is dissolved, physically dissolved, and 98.5% of it is binding to hemoglobin to form what we call oxyhemoglobin. This is very important, but it's also important to realize it's only this part, the 1.5%, that contributes to the partial pressure. Not the 98.5% does not contribute to the partial pressure. Sounds good? So let's go to carbon dioxide then. Carbon dioxide likes water a little bit more, so 10% of it can be dissolved in the plasma. But what about the others? We have 90% left. Only 30% is binding to the hemoglobin, and doesn't really bind to the heme. It binds to the carb, to the globin, and to the protein chains, not to the heme group. And so, but that's 30% only, and leaving us with more than half, 60% of the carbon dioxide is going to be transferred or transported in your blood as bicarbonate. And you're wondering, where is the bicarbonate coming from? Well, we're going to talk about that in a second, but if you remember from chemistry, CO2 plus H2O, it gives you H2CO3, and the H2CO3 will dissociate into hydrogen ion and HCO3. So if we have a mechanism to accelerate the HCO3 formation and getting rid of the hydrogen ion so it's not in our way, so now we have a lot of carbon dioxide being carried around, and we'll see the importance of the carbonic anhydrase later on. We'll see also the effect of what we call the chloride shift later on, but we know now where the HCO3 coming from, right? CO2 dissolves, giving you chemically, that's a chemical reaction. It's not just being dissolved, it's a chemical reaction. It will give you H2CO3. H2CO3 dissociates reversibly, giving you hydrogen ions and HCO3, and we will see the mechanisms that are meant for HCO3 to persist in your plasma. We'll see that. But I just want you to remember this distribution, 10%, 30%, 60% versus 1.5% physically dissolved oxygen, 98.5% bound to hemoglobin to form to you the oxyhemoglobin. Okay, so partial pressure, if you remember this equation we had here, the partial pressure, remember this is the one that's contributing to the partial pressure, right? So the partial pressure for oxygen is the one that will tell you whether or not you need to saturate. This is an important word here. Saturation of the hemoglobin. Saturation of the hemoglobin, this is the main player. You have more oxygen, then you're saturating the hemoglobin to form oxyhemoglobin. You get rid of the partial pressure, so you're consuming the oxygen, then the oxyhemoglobin is going to give up some of its oxygen, right? We call that oxygen dissociation. So in this way, it's oxygen loading, and in this way, it's oxygen dissociation. Okay. So the curve really looks like that. And we realize that the hemoglobin has 4 spots in which can bind oxygen, right? So if you bind all the 4 spots, then you reach 100%, right? Saturation or loading of your oxygen, which is over here. This, as you can see here, is close to 100%, and so we have about 90-something percent, 99% or 98%, right? In this case, all the hemoglobin is saturated. In this case, all the hemoglobin spot, 1, 2, 3, 4, is saturated. right? But this happens if the partial pressure of oxygen in the blood is around 100, from 90 to 100. You will have complete saturation, which is very good, because if you remember, this is at the lung. This is the partial pressure of oxygen in the alveoli. The alveolar partial pressure for oxygen is approximately 105. When it gets to the capillary, it's about 100, 105. So that means all the spots of your hemoglobin are loaded, and now you have saturated your oxygen. Great. Then that oxygen is going to go, if we reverse it this way, we are now talking about unloading the oxygen. So as the blood goes to the tissues, if the partial pressure for oxygen drops to about 40, right, which is the venous blood. If you remember, the venous blood was approximately, or the deoxygenated blood is about 40 partial pressure. It's going to give up, and you would think most of its oxygen. No, actually just gave 25 to 30% of its oxygen. Because if you draw a line here, you will see the line goes to about 75%. That means I lost only one of the oxygen. I unloaded one of the oxygen only of my hemoglobin. And that will mean that your venous blood, your deoxygenated blood, it's actually missing just a single oxygen, and it's requiring one more oxygen to be added there, which is really good. So you have in your blood a reserve amount of oxygen if needed, right? It keeps you going for a while, because if your partial pressure for oxygen, and please remember that the partial pressure for oxygen is the free oxygen. Remember, 1.5% of your oxygen is physically dissolved, 98.5% of the oxygen is binding to the hemoglobin. So if the partial pressure, that means the dissolved amount, that means the 1.5% of your oxygen, goes down to create a partial pressure below 40, then you get into what we call the steep phase of the dissociation. Then your system realizes that, wait a second, you are in a problem here, because even the venous, the deoxygenated blood, should not go below 40. If it goes below 40, you really need my oxygen now, because for some reason you're losing too much oxygen. So it's gonna unload about 50% or so in a very short time. If you look here, from 70% saturation or 75% saturation and 30% saturation, it took me only from 40 to 20 to unload 2 more particles of oxygen to give you very quickly additional oxygen, because now you're in trouble. Your partial pressure of oxygen in the plasma should not go below 40 in the normal conditions, but if it does, then I really need to give up my oxygen. This kind of dissociation curve is very interesting, because it shows me this very sharp descent in the saturation. So I can cough the blood very quickly to the tissues, and from the saturated hemoglobin, or the semi-saturated, from 70%, I can drop it down to 25% very quickly in order to supplement some of the oxygen if you are in trouble. The other thing is at the other end here of your saturation. You see, if in your lungs you're not having fully saturated oxygen, maybe at some point you are getting only about maybe 80, partial pressure of 80 inside your alveoli. Does that mean your hemoglobin is not going to work? No, you're actually fine. Even if your partial pressure of oxygen in the blood gets to about 80, you're still at almost 100% saturation. So you're at about 90-something% saturation. So we call that the S curve. See how it flattens here at that point between 80 and 100, right? Partial pressure 80 and 100, and you don't see much change in the saturation of oxygen, which is great. And if I even increase the oxygen concentration beyond that, if I go all the way to absolute oxygen, right? Absolute oxygen, I will not see any increase in the saturation, because a saturated hemoglobin is a saturated hemoglobin. But to the contrary, then the free oxygen will be more, because now there is no sponge to bind to it. And that will result in the free oxygen, which is the partial pressure of oxygen, being higher than 100. And by the way, that can be fatal. That can destroy your brain if you have too much partial pressure oxygen if you have too much free oxygen. So there is a very good system here to prevent overloading. Once you're about 80 to 100, that's it. The hemoglobin cannot take anymore. But it also tells you that you're not going to be in major trouble if your partial pressure of oxygen is lower than 70. You're okay. Your hemoglobin is still saturated. Yeah, you lost maybe about 5% of the saturation, but you're fine, right? So the oxygen quality in the room, if it drops about, I don't know, from about 20% or so, if it drops, remember this is of course not the atmosphere, this is in the alveolar air, but let's say the quality of oxygen in the air has dropped to 20%, right? You're fine. You're not going to die. You're fine. Even if it drops to 50%, you will be okay because you may get into about 60-70% saturation, so you will be fine. But anything below that, then you are in trouble, right? Anything, if you start dropping it 50%, then we're talking, because 50% is going to drop the atmospheric oxygen to about 70, and that's in the atmosphere. But then once you remember the old air and the fresh air, so once the atmospheric air gets into the alveolar air, then it becomes, maybe from that point it's going to be 40. That's what's inside your alveoli, and that's no good, because the 40 is the deoxygenated partial pressure, and so it's not gonna really load oxygen at that point for you. So you're in trouble if you have if you reduce the partial pressure of oxygen in the air about 50%, right? Okay, so that S curve is what we call the oxygen dissociation curve, and that's a very, very important in understanding the mechanisms of loading and the mechanisms of unloading. And they also found the following. You see how we have 4 spots here? Each spot, if you bind oxygen to it, and that's a very interesting fact, when you bind oxygen to the hemoglobin here, there will be a change in, you remember our silly rule in the lecture, a protein binding something will cause a conformational change of that protein. So when the hemoglobin here binds an oxygen, the change in the hemoglobin shape will increase the affinity of the other oxygen binding spots to bind oxygen. So one binding will allow the next one to bind, and will allow the next one to bind, and will allow the next one to bind all the way to the saturation. So it encourages the other spots to load the oxygen. And conversely, it's the same. It's the same story. I hit something accidentally here. Okay, so conversely, it's the same thing. So when you are dissociating oxygen, this one will unload, and the unloading of the first oxygen will encourage the next oxygen to unload as well. And so it facilitates the unloading. So you're helping the loading when you're going up, and you're helping the unloading when you're going down by filling or unloading of your binding spots in the hemoglobin. Sounds good? So keep that in mind, and that's what produces to us this S-shaped curve, which we call the oxygen dissociation curve. Okay, so we talked about that and in the graph, and I just want you to realize the loading and the unloading and the importance of all these events that we talked about. So hemoglobin is very interesting and it's very important. If we don't have any hemoglobin, imagine the following. You have the alveolar air here. It has 104, and it's going to go, and remember the oxygen amount in your blood is only 1.5%, right? And so, but once you reach the partial pressure that's equal to the partial pressure in the alveoli, that's it, no more diffusion. We call that equilibrium, right? But in the presence of oxygen, what happens is the oxygen, in the presence of hemoglobin, what happens is now the oxygen is going to start to be sucked into those molecules, right? Get loaded to these molecules in the oxygen loading, right? And when you load the oxygen that's in the plasma to the hemoglobin molecules, then you don't have much free oxygen. And because you don't have much free oxygen, that would encourage more oxygen to diffuse across the respiratory membrane, right? And so you keep sucking the oxygen from the plasma, the free oxygen, into your hemoglobin until you are saturated. Once you are saturated, the remaining oxygen can be in equilibrium, and that's okay. But look how much you gained. When it came to oxygen, you loaded all these things, yet you still reached equilibrium. You see how important the hemoglobin is? It's not just a vehicle. It sucks in the oxygen as it diffuses, and it builds it in, and therefore you can keep bringing in, you can keep sucking in some more oxygen through the respiratory membrane while you're loading your storage. Make sense? Good. So, once it's loaded, the oxygen, now it's going to swim, the blood is going to go with the left ventricle and the aorta and all that stuff into the capillaries. And please remember, it's the capillaries where the exchange of gases happen, right? And now you will unload your oxygen. Well, the first determining factor for the unloading of oxygen is, if you remember, the most important one is the partial pressure. Why is that important? Well, let's go in the reverse orientation now. So we are going here. This is the oxygenated blood, right? This is the arterial blood. And once it hits the tissue has been consuming the oxygen, right? And so your partial pressure in the plasma, well, first the partial pressure in the cells, and the partial pressure in the cells will affect the partial pressure in the interstitial space, and the partial pressure in the interstitial space will affect the partial pressure in the plasma, right? So now the partial pressure in the plasma has dropped, right, to about 40, right? What does that tell your oxygen that is here on the hemoglobin? To cough up some of its saturation. So this saturated molecule here is going to give some of its oxygen, and therefore you go down to 70. So the most important factor in the unloading of oxygen from the hemoglobin is the partial pressure of oxygen, right? That's the most important factor. But we will realize in a little bit it's not the only factor that allows the oxygen to be unloaded. Because face it, if the tissue is metabolically active even a little bit more, so it's producing heat like your muscles, if the tissue is producing more hydrogen ions, all of that is going to tell you, or producing more carbon dioxide, all of that is going to tell you, whoa, wait a second, we need more oxygen here. It's not all about the partial pressure for oxygen, give us some more oxygen. So you will realize that we have other factors that can play with the dissociation curve of oxygen to unload more oxygen to the tissue for you. Okay, so what are the other factors that we talk about? So here is the oxygen dissociation again, and again the main important factor here is the partial pressure for oxygen, but again that's not the only one. If you have more carbon dioxide that's at the tissue level, so you are allowing more oxygen to unload. You see the shifting happens to the right. If I have more acid, then I'm allowing more oxygen to unload. Again, I'm shifting it to the right. Temperature, that means the tissue is metabolically active, then I'm shifting it to the right. The bisphosphoglycerate, that means the tissue is depending on anaerobic respiration, on glycolysis, so I need to unload some oxygen because otherwise there is a lot of glycolysis going around. All of these factors are going to shift to the right. That means they will allow the dissociation of oxygen to happen much faster. You see here at 70 or 60, right, partial pressure of 60, initially 90% of the oxygen is saturated, right? But in the presence of high carbon dioxide, then it will be 80 or 75% of the hemoglobin being saturated. So I encouraged the oxygen to give up more, and I encouraged the hemoglobin to give up more of its oxygen just by increasing the carbon dioxide, just by increasing the acidity, just by increasing the temperature. Making sense? So these are very important factors that govern the oxygen dissociation and the oxygen dissociation curve. Keep that in mind, please. Okay, great. So here's another picture for the thing, just showing you the effect of temperature. So here we have the normal oxygen dissociation curve, and if I increase the temperature slightly, then obviously, here is the normal, rather 38 degrees, and if I increase it, then the oxygen dissociation will happen faster. If I reduce the temperature, which doesn't really happen in normal condition, you will never have your temperature being 20 or 10, the core temperature. But anyways, if that happens, then the dissociation happens at a much less rate. Similarly, the carbon dioxide, as we said, and the hydrogen ions will influence the rate at which your hemoglobin is coughing up its oxygen, saturation of oxygen, so therefore allowing more oxygen to be free and more oxygen to be consumed. Remember, the oxygen, as it's bound to the oxyhemoglobin, to the hemoglobin to form the oxyhemoglobin, is really useless to the tissue. The tissue cannot use that directly. It will use the partial pressure of oxygen, which is the dissolved amount, the free amount, that the tissues can grab. But if something is binding to the hemoglobin, they can't really utilize that. Makes sense? At least directly. Okay. So carbon dioxide on the other hand, we agreed that 10% is dissolved, 30% is binding to the hemoglobin, and 60% is as bicarbonate. We'll see something about the bicarbonate later on. So, but if the partial pressure for carbon dioxide has increased, which happens when? Think about it for a second. When do you have the partial pressure for carbon dioxide increasing? In a metabolically active tissue. Muscles, kidneys, whatever, right? That are metabolically active. So they're breathing and they are releasing a lot of carbon dioxide from the citric acid cycle, right? From the Krebs cycle. And now you have a lot of carbon dioxide, which will contribute to the partial pressure being slightly elevated. If the partial pressure goes high, then you will dissociate the oxygen faster. Remember the oxygen dissociation? We have a name for that. We call that the Bohr effect. The Bohr effect. And we'll see the Bohr effect in a little bit. But essentially, the Bohr effect will tell you that if there is hydrogen ions, if there are increased hydrogen ions, if there is carbon dioxide, that will encourage the dissociation of oxygen from the hemoglobin. That's the Bohr effect. Now, people often confuse the Bohr effect with the Haldane effect. The Haldane effect tells you if you have a lot of oxygen, if you saturate your hemoglobin with oxygen, that allows the carbon dioxide that was binding to the hemoglobin, which is 30%, to be dissociated. So it's an inverse relation. If you have higher carbon dioxide, that allows the oxygen to be dissociated. If you have higher oxygen amount, high oxyhemoglobin, that allows the carbon dioxide to be dissociated. So we have this kind of very interesting relationship between the two, which is very important. If you have oxygen in the lungs, right, and you have oxygen in the lungs, I hope so, so that will allow the carbon dioxide to get off its binding sites in the hemoglobin, and therefore it will come off in your breathing out, right? And conversely, at the tissue level, where the tissue is metabolically active, then we're talking about the Bohr effect. The Bohr effect is if you have too much carbon dioxide, too much hydrogen ions, then it will allow the oxygen to dissociate. See this kind of very intrinsic relationship between the two. Okay. So the Haldane effect tells you that the deoxygenated blood, deoxygenation of the blood increases the ability to carry carbon dioxide, and oxygenation of the blood reduces the ability of your hemoglobin to carry carbon dioxide, okay? So it's something, right, that affected the ability of, so the ability, let's summarize this instead of this confusing statement, right? So the ability of the carbon dioxide to be carried on the hemoglobin depends on the saturation of oxygen of the hemoglobin. We get that? Okay. So the more oxygen you have, the less carbon dioxide you will be carrying on the hemoglobin. The less oxygen you will have, the more carbon dioxide you will be carrying. So something, the variable here, the variable is your oxygen ability, and the result is the carbon dioxide. So you change the oxygen, resulting in changing the carbon dioxide. That's the Haldane. Once again, You change the oxygen. The result is you change the carbon dioxide. The Bohr is the opposite. If I change the carbon dioxide and if I change the hydrogen ions, then I am changing the affinity of the hemoglobin to bind oxygen, right? So oxygen carrying capacity becomes the result, not the variable. Making sense? Once again, in the Haldane effect, you change the oxygen carrying capacity or carrying ability, not capacity, carrying amount, right? The saturation. I change the saturation of the oxygen on the hemoglobin. That changes the hemoglobin ability to bind carbon dioxide. That's the result. In Bohr effect, if I change the carbon dioxide, then it changes the ability of the hemoglobin to bind oxygen. See the difference between the Haldane and the Bohr effect. It's very, very important to realize those two. Okay? And the relationship between them. Make sense? Okay. We get the Haldane and the Bohr effect. So the very last thing is, where does the carbon dioxide coming from? Okay, well, let's talk about it. Carbon dioxide, well, it's coming from the tissue. And now you're loading your tissue with carbon dioxide, but please remember that the amount of carbon dioxide that can be carried by the blood as a dissolved, physically dissolved carbon dioxide is only 10%. So you can't carry too much of the carbon dioxide, even though it's significantly higher than the oxygen. Remember, the oxygen, the amount of oxygen being dissolved in the blood was, do you remember? 1.5%, right? Here we had 10%. Amount of carbon dioxide that will be carried on the hemoglobin molecule will be 30%. It still leaves us 60% as the bicarbonate. So where is the bicarbonate coming from now? So let's talk about that. So tissues here, this is metabolically active tissue. So it's unloading a lot of carbon dioxide. So I have a lot of carbon dioxide, but remember, carbon dioxide doesn't dissolve much. So we have some of it will dissolve, right? But 30% will bind to the hemoglobin, giving us this reduced hemoglobin, right? But really the main part is being carried as bicarbonate. And this is the tricky part here. We need to realize that also, right? So where is this coming from, the bicarbonate? We're going to realize that carbon dioxide and water, in the presence of an enzyme, very important enzyme, very, very important enzyme, called carbonic anhydrase. Carbonic anhydrase. The next time we talk about this carbonic anhydrase will be in your kidneys. So inside the red blood cells, we have carbonic anhydrase, and the carbonic anhydrase will catalyze this reaction. Instead of going to carbonic acid, and the carbonic acid dissociates into hydrogen ions and bicarbonate, no, we are taking a shortcut. So CO2 plus H2O will give me hydrogen ion directly in the presence of carbonic anhydrase and HCO3. The hydrogen ion is capable of binding the hemoglobin, giving me reduced hemoglobin. Right? Giving me a hydrogen ion, not reduced. Reduced hemoglobin is the hemoglobin without oxygen. That's reduced hemoglobin. So ignore that last word. So it's capable of binding the hemoglobin. The hydrogen ion is capable of binding to the hemoglobin. But also remember the following. Remember what happens when hydrogen ion binds to the hemoglobin? The hemoglobin has less chance to bind oxygen. The same thing here. When carbon dioxide binds to the hemoglobin, that hemoglobin now has less chance of carrying oxygen. Remember what we call the effect that governs those two events? Bohr effect. Bohr effect is if carbon dioxide binds hemoglobin, or if hydrogen binds hemoglobin, then the oxygen carrying capacity of the hemoglobin is reduced. So that, just keep that in the back of your mind. So let's go back to the equation here. Carbon dioxide plus hydrogen, giving you, in the presence of carbonic anhydrase, it gives you a hydrogen ion, which gets sucked by the hemoglobin, and the HCO3. So now I'm making a lot of HCO3, and I want the HCO3 to get out of my red blood cell into the plasma. So I need to exchange it with another anion, right? what is the most abundant anion in the plasma or the extracellular fluid? We talked about that when we talked about the action potential. Chloride, right? Chloride. So the chloride can get in the cell and the HCO3 can switch out of the cell, right? We call that chloride shift. Chloride shift. We have tons of chloride. It's not a problem to give some chloride to the red blood cells. In exchange, we have HCO3 coming out. That's the source of your HCO3. Remember, 60% of your carbon dioxide is being carried as HCO3, which is made thanks to the carbonic anhydrase inside your red blood cells. And then the HCO3 that resulted from this reaction is going to be switched by the chloride, right? Exchanged with the chloride in something we call the chloride shift. Chloride shift. And now you have HCO3, and when we get to the regulation of pH of the blood, you will realize that the HCO3 is very, 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 very critical for maintaining your pH, your normal pH. So keep that in the back of your mind, and now you understand the carbonic anhydrase, you understand the chloride shift that happens between the HCO3 and your chloride ions. Okay, so what happens then in the lungs, right? What happens in the lung is that oxygen comes in. Why does it come in? Because the partial pressure of oxygen here is much higher than the partial pressure of oxygen there. So now I'm loading it with oxygen. Conversely, I'm unloading the carbon dioxide. Why? Because the concentration of carbon dioxide here is much lower, well, slightly lower. 46 here to 40 here. So I'm unloading the carbon dioxide. Sounds good? But let's talk about this a little bit. When oxygen binds to the hemoglobin, right, it also has the capability now of dissociating the carbon dioxide. Remember? We call that, so the variable now is the oxygen and the result is the carbon dioxide. We call that Haldane effect. So that means the carbon dioxide now is going to come out of the hemoglobin and therefore will increase the ability of the carbon dioxide to keep shifted into your lungs, right? Also, when you're increasing the hemoglobin, when you're increasing the oxygen concentration on the hemoglobin, now the hydrogen ions also dissociate. And when the hydrogen ion dissociate, it's going to bind together with the HCO3, and that will reduce the concentration of HCO3 inside your red blood cells. And if you reduce the HCO3, then fresh HCO3 from the plasma, it's going to exchange itself with the chloride ions, right? And so now you have new HCO3 coming from the plasma to replenish the HCO3 that you will be losing. Where is the HCO3 here going? Well, again, in the presence of carbonic anhydrase, HCO3 will combine with hydrogen ion to produce to us CO2 and and water. Well, the CO2 is going to go out again in the lungs. So now I have loaded the oxygen, and I unloaded two things. I unloaded the hydrogen ions that I was carrying around, and I unloaded the carbon dioxide. Very important for us to understand and remember this graph here about the exchange of oxygen, exchange of carbon dioxide, both the events that happen at the tissue level and also the events that happen at the alveolar level. Very, very important for us to understand those two events. Sounds good? Okay. So that pretty much summarizes to us the physiology of the exchange, the gas exchange, and the carrying of gases, which we call gas transport. So I wanted to make these two videos in case we don't get into much details in the lecture talking about them, because I would like to have some time for us for lab activities. And so, but at least now you have reference that hopefully will allow you to review this material. And for all the guests, I hope that this will help you studying as well. So thank you for watching this, and I'll be making more videos, so please stay tuned and have a great day.